صباح الخير طلاب. أنا دكتورة نبراس سليم العمار. راح أكون وياكم محاضرات الإميونولوجي. Our lecture for today is immune response part 1. And next week we are proceed in part 2 of immune response. Please, Arthur. طبعا مطلوبة من عندكم هذه definition are basic tools of immune system. How immune system deal with bacteria and viruses in extracellular space, dealing with intracellular pathogen, and how does the immune system being alert to the presence of infectious agents inside the cell. And we will mention these terms, antigenicity and immunogenicity. طبعا دكتورة دانيا already mentioned innate and adaptive immunity. مو صحيح؟ Now, basic rules of immune system. First of all, recognition. The immune system have ability to recognize what is foreign, non-self from the self antigen. In that way, the critical role here of the immune system is to determine, as we say, what's foreign and non-foreign bodies or molecules. Therefore, the cells and molecules of the immune system have that comprise the innate immunity. You already knew what is the innate immunity. Preoccupied. That's me. It's pre-educated. What is by detecting the presence of plants? What is plants? What are the plants? Plants are pathogen-associated molecular partners. That means the molecules that present on the foreign. For example, on the pathogen, bacteria, on virus, on any foreign molecule. So the immune cells and molecules of the immune system preoccupied already know how to build plants. In this figure, the pathogen associated with plants, that means that any foreign pathogenic bacteria or virus or any foreign substance will here on its surface some molecule, specific molecule that we will recognize by the immune system. As we say, we call these molecules PAMs, the pathogen-associated molecular patterns. These plants will be recognized from by different mechanisms. Will be recognized by pattern recognition receptors, PRR. These pattern recognition receptors may be soluble, present normally in the body, like, for example, toll-like receptor, complement, some pathogens. Also, there is some association. There are On the cell of immune cell, there are these receptors associated with the pathogen-associated molecular pattern and will proceed in elimination of these foreign substance or pathogen. Now, in soluble PRR, binding of microorganism by soluble, as we said, PRR lead to enhancement of phagocytosis. Already knew the pathway of phagocytosis. You know what is phagocytosis? So proceed in that catalytic cascade that lysis. Also, when the PRR binding comes on the surface of immune cells, also proceed in phagocytosis and then activation of immune system and release of cytokines and chemokines that amplify the immune response in that case. طبعا لو ما قاعدين طلاب يعني هسه موجودين انتم كانت المحاضرة مكان لكم كلكم. Now the next step is elimination. As we said previously, elimination by phagocytosis. Is initiation of memory, immunological memory. Immunological memory will be present as a result of that because the immune system is able to keep observe cells and molecules that have ability to remember. Remember, this is pathogen, this is specific pathogen. And will be specifically active in the second exposure to this pathogen. So bacteria or virus, we call it immunological memory. In that case, we proceed first. In the first recognition, we are talking about innate. Is that true? And then here we are proceeding in adaptive. Okay? Also, another important issue is the immune regulation, immune checkpoints responses, in order to make sure that the immune response is suitable to the treatment. I mean with this agent. It's suitable. To eliminate this threat. Also, to make sure that these checkpoints make the immune system make sure that there is no response toward cell. Only toward non-cell. Only toward the pathogens or foreign substances. Immune response stop in that case. Why this immune response equal? Can we say it if it's proceed the immune response? Yes. make injury to the tissue, nearby tissue, or the cells of the body. Yes, very good. Now here in this diagram, there are different difference between mechanoapoptosis and in case of necrosis. For example, in apoptotic cell, as you see here, the physiological stimuli cause mild injury, and the cell program to death. I mean, for apoptosis program cell death. In that case, it will go during through a regulating its death by exposed to the immune system to mark the cell for the immune cell. يعني يسويها tag or mark in order for immune cell to recognize this apoptotic cell in structure of the cell. شنو المهم بيها؟ Why this is important? Because going into a apoptotic process, there are, as we see here, DAMs, danger-associated molecular data. These DAMs, if the immune system recognizes these DAMs, there will be immune response. During the apoptosis, these DAMs stay hidden inside the cell because it will not rupture. It will be tag itself to the immune cell, for example, phagocytic cell to engulf B cell and phagocytosis in order not to expose the DAMs to the immune cell, other immune cells, because it will initiate another immune response, maybe lead to injury. Okay, once recognize the mark or the tag on the cell after cell and phagocytosis occur. War or any injury, any trauma, anything cause some harmful effect to the cell, it will be necrology process. In case of necrotic cell, the plasma membrane will be swollen and rupture. After rupture of plasma membrane, the DAMs, danger-associated molecular pattern, will be released occur, and immune response will start when these DAMs will bind with RR, will bind with these DAMs and stimulate the immune response. اوكي واضح؟ ماك سؤال؟ Now dealing with bacteria and viruses in the extracellular space. For both in another lecture راح اشرحها لكم incoming lecture in that case. The complement, toothpaste protein, antimicrobial peptide. Then remove them by destroying them through the membrane lysis. هذه راح in details هذه المحاضرة. The same classes of protein on board in decorating the pathogen or infectious agents, bacteria or virus for recognition for phagocytosis. يعني هي تسوي decorating للباثوجين، يعني bind with the pathogen as service marker in order to be recognized by phagocytics. So we observe these pathogens for eating, or not eating. We call it opsonins. Opsonins. So we have steps: phagocyte recognition by dams and opsonin will bind together. We have next slide. I will explain it more effectively. Engulfment, degradation of the pathogen occur in that case. The role of phagocyte, you already knew what is phagocytosis. الدكتورة دانيا شرحت لكم كمان عيدها بس مجرد خليها للقراءة من بعد. We have many phagocytic cells. Types, many types هذه قسم منها not all. Macrophage here present in all organs, ingest and destroyed microbe through antigen to T cell by immunity, produce cytokine and chemokines. Monocyte present in blood 5 to 7% and recruited out infection site and differentiates into macrophage. As you know, monocyte is one of the phagocytic cells in order to make phagocytosis. Neutrophils, when you see pus in the site of infection there, that's mean there is neutrophil. Keep this in your mind. Present in blood 60%, in blood as blood leukocyte, increase during infection, recruited by chemokines to the site of infection and ingest and destroy biogenic bacteria, for example, Staph aureus and Streptobiosis. Now, I want to show you how the PAMS and PRR will be bind together and how opsonin bind and attract phagocytes. Here in this figure, you can see the microbe. Each microbe has specific pathogen-associated molecular pattern. That means have some, for example, antigen, some molecules on its surface. يعني مثلا bacteria, for example, have some antigen, somatic flagellar antigen or capsular antigen. يعني تخيلوها بهالشكل. When PAMS bind with PRR and in other side the opsonin like complement, for example, antibodies or acute phase proteins. These bind on the pathogen because it will note that this is foreign substance or pathogen. So when opsonin bind on the pathogen and then it has each opsonin has specific receptor on phagocytic cell. For example, the complement have a receptor on the phagocytic cell. يعني إلى receptor allophagocytic cell will bind to the complement and then proceed in that case in phagocytosis. The opsonines include complement proteins, C3b, C4b. As I mentioned, I will explain it in more details in complement lecture. Antibodies like IgG, IgM, and acute phase proteins like C-reactive protein. C-reactive protein, you can refer to key definition in order to know what is the definition of all these terms. Mannose binding lectin, also one kind of opsonine. واضح؟ ما في سؤال؟ Now, dealing with intercellular pathogen. When the virus or some parts of bacteria enter inside the cell. Now, how a new cell deal with this situation? The protein and phagocytic cell no longer be of any use of dealing with this type of infection because the protein cannot penetrate inside the cell. That's true. And also the phagocytic cell cannot deal in that case with in this kind of situation, although some specified phagocytes like microphages can kill intracellular bacteria. That means ممكن الphagocytic cell when bacteria inside the phagocytic cell have a way to kill the bacteria inside the cell. But many bacteria can escape this mechanism. Not all bacteria can be interested inside the phagocytic cell. أفتحوها بالmicrobiology. شوفوها بالlecture of microbiology. There are some species of bacteria can escape phagocytosis. Here, we mentioned that phagocytic cell can kill bacteria inside the cell by this mechanism, either by or oxygen independent pathway. أخذتوها دكتورة دانش حكيتها؟ لا شرحتها لكم؟ لا ما شرحتها؟ نشرحها يعني؟ إيه لا يعني ما مارين بيها بس تاخذ الوقت. Phagocyte intracellular killing mechanisms by two ways. In the presence of oxygen and the absence of oxygen. In presence of oxygen pathway, we call it respiratory process. النفحة الهوائية يعني إذا أتوقع هون مثلا hydroxylicals, free radicals like كل all these were called pathogen in that mechanism. Oxygen independent. In the absence of oxygen, there are many proteins or factors that play in that case, like lysozyme, lactoferrin, transferrin. How does the immune system? As we said, the infectious agent now inside the cell. How does the immune system act? into the presence of this pathogen. If your immune system knows there is pathogen inside the cell, this is a very important point. There are a number of intracellular PRR, pattern recognition receptors inside the cell detect the production of signals. What are these signals? It's present in key definition. You have to key definition in order to know the definition of cytokine and chemokines that serve in communication between cells, immune cells, and alert the immune system that there is a pathogen inside this cell. Okay? There is a clever way to display the breakdown peptide outside the cell product. يعني خلي أشرحها لكم in more details when phagocytic cell, for example, or the pathogen enter inside the cell. It will break the breakdown. It will ingest this pathogen. راح يسوي له digestion. راح يسوي small peptide. This small peptide will be bounded with NFC class 2, for example. If the pathogen is bacteria, it will bind with NFC class 2 major histocompatibility complex and show it on the surface of phagocytic cell, or antigen presenting cell we call, because it presents antigen توضحها للimmune cells. The immune cells will detect the presence of any mechanism, several mechanisms by T cell, CD4 T cell in that case. We will mention it in details in the next lecture. And that's how the immune system alert in that case there is a pathogen inside the cell. واضح هذا؟ إذا شيء واضح قولوا يعني. ممكن نعيده أو نحكي بالعربي أو in more details. Okay? ماشي يعني؟ Here, like this. Displaying of breakdown product by MSC, the immune system. For example, here, dendritic cell. هذا الرسم خليته بحيث هو بالحقيقة مفصول عن تقدر ترسم يوضح التو. هو بالحقيقة كل واحد إذا فيروس هذا البكتيريا، إذا بكتيريا هذا البكتيريا تقريبا. فإذا if the antigen presenting cell, like for example dendritic cell, display the راح يصير digest the microbe, for example bacteria، راح يصير digestion إلها. small peptide راح تتكسر إلى small peptide and bind with MSC class 2. Should this peptide, which type of cell recognize this? T helper antigen presenting cell راح يصير فيها وأكو second signal. The variant is TCR with CD4 T cell with MSC class 2 peptide complex وأكو other signal اللي نشوفوها CD28 with B7 on the antigen presenting cell. Where what These two signals will result in activation of T helper T cell, CD4 T cell, and proceed in proliferation, activation, and differentiation into T helper cells. T helper cells will help other immune cells in their activities, like B cell مثلا. B cell هتسوي لنا immunoglobulin antibody against this bacteria or pathogen. واضح؟ While in the other side, if the cell is, for example, viral infected cell, يعني cell الفيروس سوالها infection inside the cell. هذه الحالة, as you know, the virus will use the machinery, genetic machinery of the cell, يعني DNA مالتها, فراح يكون البروتين اللي يتشفر هو جزء منه من الفيروس. واضح؟ In that case, the peptide, breakdown peptide from this cell will contain something from the virus, يعني foreign هو. فراح البرزنتيشن يصير with MHC class 1, major histocompatibility complex class 1, and will be and also there is second signal, second set of receptors لازم يكونون and will recognized by cytotoxic CD8 T cell. In that case, or tumor cell, إذا cell بها tumor, the same mechanism. In that case, we need to kill this cell. So the pathway is toward the cytotoxic T cell in order to kill the same cell in that case. واضح؟ واضح؟ You have, I think you know what is antigenicity and immunogenicity, بس مجرد للإعادة يعني تفيدكم إنه to remember these terms. Antigen, any foreign molecule, non-cell, can react with the product of immune response and not an immunogen molecule that induce or activate the immune response. It's very important to remember that not antigens are immunogen. واضح؟ خلوها بعدكم هذه. الimmunological terms مثل related to antigen أتصور أخذتوها دكتورة دانيال، ما شرحت لكم جزء بس حبيت أعيدها حتى مجرد تتذكروها. اتفقنا أنا وياها يعني في هذه الحالة. Epitopes antigenic determinant are small chemicals put on the antigen molecule to elect and react with an antibody. الهابتين, what is the hapten? واحد يعرفه؟ Define what's hapten? مو أخذتوه please last week؟ لو ما أخذتوه ما قريتوها؟ حاولوا كل محاضرة تقروها. تسألوا بالكلاس أي طريقة تسألون، لا تحاولوا تراكمون المحاضرات. Hapten, small molecule that is not immunogenic until it binds with other protein in order to elect the immune response. Adjuvant واحد حتى لو يقرأ كذا interactive يعني شوي. يلا واحد من عندكم يقرأ وهو قاعد مكانه يقرأ السطر موجود على هذا. يلا شنو اسمك؟ محمد؟ يلا محمد صوت عالي حتى لو تنفع هاي. Very good. شكرا. Now, the feature of immunogenicity, foreignness. It's very important that antigen is foreign, non-cell. Molecular weight high molecular weight are good immunogenes. الchemical والcomplex إذا structure مالته complex, more complex molecule have different epitopes. راح ناخذها هذه details بالانتيجين انتيبودي interaction. راح اشرح لكم كيف تكون هذه مؤثرة effective in that case. It does important to increase or decrease the immunogenicity of the antigen. Factor contribute to immunogenicity: genetic factor, age, immunosuppressed patient, lifestyle, type of diet, environmental factors. نعم، خلصنا محاضرتنا. Thank you for attendance. إذا أي سؤال، أي شيء ما فاهمين، أنا حاضرة. تفضل. واضح المحاضرة؟ واضح. بس من الأسبوع الجاي أريد interaction رجاءً. أنا أصير برود، أنا من برود أحكي وأصير أكثر بعد برود. أريد أنتم تحركون الكلاس، وجودكم يعني كلش مهم. لا صورة. شكرا جزيلا.